friends, welcome back to my channel. So today I'll be talking about cancer, which is a very popular disease, and very soon it's going to be a very common disease in every family of the universe. So what is cancer and how this cancer occur within our body? So cancer simply means it's an uncontrolled cell proliferation. That is, there is no restriction, no limit, no inhibition in cell division process. And in our last video I told you there are so many proteins which are responsible for cell division or the cell cycle regulation. The cyclin, CDK, there are so many replication enzymes which are when altered leads to cancer. So cancer is a genetic disease, but most of the cancer are non-inheritable in nature. That means they do not inherit from one generation to the next generation. Why? Because most of the cancerous gene lies within the somatic cells, not the germline. So majority of the cancer are restricted within that individual, not to inherit into the next generation. But some cancers are there which are genetic in nature, that inherit from generation to generation, and that is almost 5% to 6% of total cancers. So there is a confusion between tumor and cancer. Are they same or different? Now tumor means when a cell is growing abnormally, no longer regulated by the growth factors or the anti-regulatory factors, and expand the tissues or expand the cells and leading to an abnormal bulging structure which is known as tumor or the neoplasm. So tumor is a growth of the cells abnormally, but these growth either restricted within a localized region or it can be spreaded in nature. So tumor are of two types. One is called benign, another is called malignant. Now in case of benign tumor, the growth is limited, definite, and the tissues or the cells are not able to invade the healthy tissues. That means they are restricted within a specific region. They are unable to invade the healthy tissues. These tumors are called benign tumor, and they can be treated by surgery or medicine. But whereas in case of malignant tumor, there is a special feature which is known as metastasis. Now what is metastasis? In case of malignant tumor, the tumor cells or the abnormal cells are not restricted within that localized region. It invades the healthy tissues. Either through blood circulation, it reaches to the distant place and invade the healthy tissues, and in this way it can spread it throughout the body, and which is very dangerous, very severe, and even the most of the cases it is life threatening too. So the malignant tumors are known as cancer. So all tumors are not cancerous, but definitely all cancers are tumorigenic in nature. These cancers are of different types depending on their tissue origin. Based on tissues, cancer are of four types. First one is carcinoma. In this type of cancers, the cancer cells are made up of epithelial cells, which are ectodermal or endodermal in origin. And mostly this type of cancer are skin cancer, breast cancer, etc. Another one is sarcoma. Now these type of cancers are made up of connective tissues and which are mesodermal in origin. Example, bone cancer or cancer in muscle. Next one is lymphoma. Now in this type of cancer, there is a excessive growth of lymphocytes, lymphocyte growth, and it's mostly occur by the spleen or lymph node. And this type of cancer is present. There is a disease known as Hodgkin disease. In this case, the lymphocytes grow rapidly. And the last one is called leukemia, which is also known as blood cancer, when there is an excessive neoplastic growth of WBCs. White blood cell while growing excessively, that leads to a leukemia, or you can say it's a one kind of blood cancer. So there are 4 types of cancer on the basis of the tissue origin. Now, when a normal cell is converted to a cancer cell, there are so many behavioral changes occur. Now, in cancer cells, there are 6 fundamental features are altered. Say for example, this is a cell, and within this cell, there are 6 fundamental alteration occur. The first one is the insensitivity to anti-growth signal. That means there are so many receptors in outside the cell, and these receptors are responsible for the growth regulation. There are so many anti-growth factors which control the cell divisions, but this cell is not able to receive any kind of anti-growth signals. It's completely insensitive to the anti-growth signal, so it can proliferate uncontrollably. The second one is very important point. It's a limitless replicative potential. Now you all know, when a cell is preparing for division, the replication is very important. Replication of the chromosomal material is very important. So, and that replication is regulated by a series of controlling proteins, enzymes, and those proteins are continuously synthesized without any signaling molecules and promote the cell division process. So the limitless replication potential indicates that cell is growing uncontrollably without any restriction. Third one is angiogenesis. Now, for the growth of tumor, there is a very important essential factor which is known as the growth of blood vessels. If the blood vessel does not grow, so that leads to the restriction in the growth, or that leads to the restriction in the proliferation. But during the cancer, the growth of blood vessel is much more high and it promotes the tumor growth. So angiogenesis, that means the growth of blood vessels, the vascularization process is very fast. The fourth one is self-sufficiency in growth signal. That means the cancer cell does not require any growth signal. It is self-sufficient to grow by itself. It does not require any growth factors or any growth proteins which promotes or stimulates the cell to grow or to divide. So that means the self-sufficiency leads to uncontrolled cell proliferation. And the fifth one, tissue inversion and metastasis. It's a very important thing that cancer cell does not restrict its growth within a localized region. It invades the surrounding tissues or healthy tissues, and which are very much severe and life-threatening for most of the cancer, in most cases of the cancer. So this tissue inversion leads to the spreading of the cancerous cells throughout the body. And the last one is very important, evasion of apoptosis. Now, when there is a system in our body, when any abnormal cells are found within the cell, so body is always trying to discard or eliminate that cell via the apoptosis or the cell death process. But in case of cancer cell, this is immortal in nature. This cell inhibits its own apoptotic mechanisms and grows uncontrollably, because the apoptotic mechanism is not applicable in case of cancer cells, because the apoptotic genes are mutated and that are unable to recognize or restrict the growth of these cells. So these are the basic features when a normal cell become cancerous. Earlier I have said cancer is regulated by a series of genes. There are so many genes which involve during the cancer, because there are so many genes which are involved in the cell cycle process too. So cancer mechanism is controlled by 3 categories of gene. The first one is the Proto-Oncogene. Second one is the Tumor Suppressor gene. And third one is the caretaker gene. Now these genes, when not in mutated state, that means when it is wild type, they control the cellular divisions, cell cycle, and also the genome of our body. Now firstly, the Proto-Oncogene. These genes are normally regulate the cell growth and proliferation. There are so many genes that acts as a proto-oncogene. Say for example, anti-apoptotic gene, cell cycle CDK cyclines. There are so many genes which are responsible for the cell cycle mechanisms. They are all under the proto-oncogene. Another one is called tumor suppressor gene. By name you can understand tumor suppressor gene. That means it can suppress the abnormal growth or abnormal tumors. And these genes are responsible for suppression of tumor. There are so many tumor suppressor genes present in our body. Among them, 2 important genes is called P53, which is also known as guardian of the genome, and PRB, that is the retinoblastoma. This is also a tumor suppressor gene. The third group is called caretaker gene. These genes are responsible for DNA repair and damage recognition. So all those genes which are involved in DNA repair mechanisms are called caretaker genes. Now these genes are the normal wild type genes present in our body. But when these genes are mutated due to several reasons that I will discuss later on, when they are mutated, their function is altered. So first, when the proto-oncogene is mutated to oncogene, normally the proto-oncogene is responsible for the cellular growth and proliferation, but the oncogene, normally in case of mutation, what actually happened, the function of the gene altered. The opposite function it exerts. When a capital gene is converted to a small t, the tall become dwarf, just like that. But here, when a proto-oncogene is converted to oncogene, its function is more pronounced. That means it is also called the gain of function. and it's a dominant mutation. And what is its function? It promotes heightened level of cell division or cell growth. That means without any restriction, it promotes the cell division process. That means the uncontrolled cell proliferation. So normally proto-oncogene is responsible for the cell proliferation, but in a controlled fashion. But oncogene, the same thing it will do, but in an uncontrolled fashion. So that's why it is called the gain of function. The another one is called tumor suppressor gene. Normally it is responsible to suppress the tumor. But when it becomes mutated, its function becomes reverse. That is the loss of function. That means loss of function. That is, it is unable to suppress. So, tumor growth is promoted. Now, in case of caretaker gene, as it is responsible for the DNA damage recognition and DNA repair mechanism. That means when there is a DNA damage, so it is actually trying to prevent the damage and try to repair that DNA segments. But when it is mutated, that leads to the cell with having that abnormal DNA undergoes cell division process and leading to the abnormality in the successive divisions. That actually lead to the abnormal cells and it undergoes uncontrolled cell proliferations. Now the oncogene are of two types. One is called cellular oncogene. That is called CONC. Another is called viral oncogene. That is called VONC. Now the cellular oncogene means those oncogenes are present in our host cell. That means all the genes which are present in our body as a proto-oncogene form, say for example the growth factors, the anti-apoptotic genes, those genes when converted to the oncogene, they are called the cellular oncogene. But the viral oncogene means those genes are present in the viral DNA or the viral RNA. And those when infect into our body leads to cancer. So this viral DNA or RNA containing or possessing genes are called the V oncogene, and most extensively studied viral oncogene is present in RSV, that is the Roa sarcoma virus. Now the another important thing is how this kind of alteration takes place in our body. That is, when a normal cell is converted to the cancer cell, what is the reason behind that? There are so many reasons behind the alteration of a normal cell into a cancer cell, but the most importantly, quote unquote, you can say our lifestyle and the pollution is mainly responsible for these kinds of disease. Actually, day by day we are advancing with the technologies, and those technologies actually are welcoming these kinds of disease in our country or in the universe. So there are so many reasons behind that. We can enlist those reasons in this diagram. So let's see. Now in this diagram, I have mentioned 7 molecules which when altered leading to abnormal cell proliferations. Serially, if we say, the first is the signaling molecule, that is the growth factor, different kinds of growth factors when altered resulting into a cancer. First thing is the signaling molecule when it is mutated or converted to some different forms that is unable to regulate the cell proliferation mechanism. The another thing is the signal receptor molecules. As we all know, the cell signaling system is very important for the activity of a cell. For each and every activity, there is a cell signaling mechanism. When the signaling receptors are altered due to the mutation, so that leads to some abnormal signal, or the signal may not be transcribed properly into the cell, leading to the abnormality. The third one is the intracellular transducers, which are responsible for the activating the second messenger systems. If these transducer molecules are somehow altered or changed, so that give some abnormal signal to the cell or to the nucleus. And as a result of that, the transcription factor, the fourth one, which is also a significant part, and these transcription factor, when activated or promoted, that leads to the activation of the transcription and the translation process. So when this transcription factor gives some abnormal signal, resulting into this secretion of some abnormal proteins, and those proteins might be DNA repair proteins, they are cell cycle protein, they are the apoptotic protein, all those proteins when become abnormal, that leads to cancer. Now the question is how this abnormality takes place. The genes which are responsible for the synthesis of these kinds of molecules, when altered, become abnormal. Now, what are the mechanisms behind that are responsible for this conversion of the proto-oncogenes into the oncogene? So there are basically three mechanisms which are responsible for the conversion of proto-oncogene into oncogene. First is point mutation. Now, mutation is a very possible cause behind cancer, and mutation occur due to different reasons. There are different type of mutagens present in the environment, as well as in different kinds of food also. So these mutagens, they are either physical mutagen or the chemical mutagens, the UV radiations, the X-ray, the gamma rays. There are different type of radiations are there which are responsible for causing the base level changes, and these base level changes or the DNA level changes or the nucleotide level changes leading to the point mutation and alteration in the amino acid sequences that are responsible for causing cancer. The second one is the chromosomal abnormalities. And these chromosomal abnormalities includes the deletion, duplication, inversion, and the one of the important is translocation. That means the exchange of the chromosomal segments between two non-homologous chromosomes or the homologous chromosomes. So that deletion, duplication, inversion or translocation, that leads to the change in the chromosomal configuration. And these abnormal chromosome give some abnormal signal to the cell and leading to abnormal or uncontrolled cell proliferation. And the third one is another important thing is the amplification of DNA. And there are series of enzymes which are responsible for the DNA replication. So when those enzymes, without any control, without any regulatory factors, undergoes continuous replication process, that I told you previously, that is limitless replication potential, and that leads to the synthesis of a huge number of DNA, huge number of the apoptotic genes, and that overpopulation of these apoptotic genes or the proto-oncogenes leads to the cancer. So there are three possible causes behind the cancer. Now we come to the last part, but not the least one, that is treatment. In cancer, there are no such definite treatment right now present apart from the chemotherapy and the radiotherapy. And these therapies are applicable and save the life of the patients when the cancer is diagnosed at earlier stage, that is grade 1 or grade 2. But when it is in grade 3 or grade 4, there is very low percentage of chances to be survived using these kinds of treatment. But right now there are different type of strategies are adopted, but those are under the trial process. In future we will expect some better treatment procedures which are under right now trial process, and these processes are mainly immunotherapy. There are two types of immunotherapy. One is the passive and there is the active. And in this therapy, in this active therapy, the host immune defense mechanism is taken into consideration, and this host defense mechanism participate as a treatment measure for the cancer. Whereas in case of passive therapy, some antibodies or some proteins which are injected into our body to treat the cancer. In second one is the Antisense RNA therapy. And in this therapy, the main purpose is how to inactivate all the cancer causing proteins or the cancer causing gene. That means the purpose is inactivating cancer causing genes or the proteins. The next one is the prevention of angiogenesis. That means the growth of blood vascular system, the growth of blood vessels are restricted in this treatment procedure that actually limits the growth of tumor cells or the cancer cells. So these are the under trial method, and in later future we will expect that some better strategies or better treatment mechanisms will be coming and cancer become a very normal disease then. So in this video I am just describing the basic mechanism of cancer. What is cancer? What are the reasons behind cancer and the genes responsible for cancer? In the later video I will discuss about the genes, mainly the tumor suppressor genes, the P53, the PRB, how these genes regulating our normal body system, and where it becomes mutated, what is the effect of those genes.